You begin as a perfectly normal, law-abiding epithelial cell inside a human tissue wall. Every day, you follow instructions, repair your structure, and look out for your neighboring cells. But today, something breaks. A random copy error during routine cell division, a stray particle of ionizing radiation, or a chemical carcinogen slips past your defenses.
Your DNA snaps. Specifically, your proto-oncogenes, the genetic gas pedals for growth, get permanently stuck in the on position. Simultaneously, your tumor suppressor genes, like the regulatory p53 gene, are completely knocked out. The internal checks and balances fail. You don't feel any different yet, but you have just broken the fundamental code of multicellular life. You have stopped listening to the body. Welcome to the mutation.
Normally, a cell with this much genetic damage is ordered by the immune system to commit suicide through a clean process called apoptosis. You receive the molecular suicide signal, but you ignore it. You activate an enzyme called telomerase. In a normal cell, telomeres act like a biological countdown clock, shortening with every division until the cell naturally dies of old age. By turning telomerase on, you rebuild your telomeres indefinitely. You have unlocked cellular immortality. While your neighboring cells grow old, wear out, and fade away, you remain perpetually young, unstable, and ready to divide. You begin to divide. One cell becomes 2, 2 become 4, 4 becomes 16. You ignore the tissue's structural borders and contact inhibition, the natural signal that tells cells to stop dividing when they crowd each other. Your rapidly expanding family forms a dense, localized cluster of chaotic cells known as hyperplasia. The tissue layer begins to warp. On the outside, the host doesn't feel a thing. There is no pain, no fever, no swelling. But inside, you are quietly establishing a rogue colony, outcompeting the healthy, functioning cells around you for basic glucose and oxygen.
As your colony expands into a solid, distinct tumor, you hit a hard, physical wall. You have grown too large for the local blood supply to sustain you. The center of your mass is suffocating from a lack of oxygen, hypoxia. You are starving. You flip the angiogenic switch. Your cells secrete massive amounts of chemical distress signals, primarily VEG, vascular endothelial growth factor. These chemicals drift through the tissue and hijack nearby healthy blood vessels. The blood vessels sprout new, chaotic, leaky branches that twist away from their regular paths and drill straight into your core. You have successfully rewired the host's plumbing to feed your empire.
With an unlimited supply of oxygen and glucose pouring through your new vessels, your growth explodes. You form a fully developed, localized tumor. Biologists call this stage Carcinoma In Situ. You are highly abnormal, packed tightly together and completely disorganized, but you are still contained. You are sitting on top of a tough, fibrous barrier known as the basement membrane. This membrane is the only thing keeping you separated from the rest of the body. Doctors run a routine screening or a blood panel. They might see a slight elevation in marker proteins, but you are still a hidden local threat. The localized boundaries are no longer enough. You need to expand. You undergo an epithelial mesenchymal transition, EMT, a radical transformation where you shed your rigid shape, detach from your sibling cells, and become highly mobile and aggressive. You deploy a battery of destructive enzymes called matrix metalloproteinases, MMPs. These act like chemical drills, melting away the tough basement membrane that held you back. You crawl through the dissolved tissue matrix until you reach one of your newly formed leaky blood vessels. You squeeze your way between the endothelial cells and drop straight into the rushing torrent of the bloodstream. This process is called intravasation. You are officially in circulation.
The bloodstream is a violent, lethal highway. The mechanical shear force of the pumping blood threatens to tear your cell membrane apart. Worse, the host's immune system detects you. Natural killer cells and patrolling T cells swarm your position, recognizing you as a dangerous anomaly. You adapt immediately. You secrete clotting factors that attract blood platelets, wrapping yourself in a dense, sticky shield of the host's own clotting proteins. You are a wolf in sheep's clothing. The immune cells cruise right past you, unable to see the malignant threat hidden beneath the blood clot. You survive the transit, floating through the vast arterial network.
You catch on the microscopic walls of a distant capillary bed, perhaps in the liver, the bones, or the brain. The vessel is too narrow for your platelet shield to pass. It's time to exit the highway. You use specialized surface adhesion molecules to anchor yourself firmly to the inner lining of the blood vessel. Once stuck, you deploy your MMP drills again, eating a hole through the vessel wall and crawling out into the lush, uncharted territory of a completely different organ. This is extravasation. You have successfully landed your first scout team on a foreign shore.
Most cancer cells die at this stage, unable to survive in the unfamiliar chemical environment of a new organ. But you are resilient. You secrete specific growth factors that manipulate the local stromal cells, forcing the new environment to adapt to you. You begin to divide again, forming a secondary tumor site, a metastasis. The host starts to feel the systemic weight of your multi-front war. They experience unexplained weight loss, cachexia, chronic fatigue, and organ-specific failures. If you are in the bone, the bones weaken. If you are in the brain, neurological deficits begin. Oncology teams launch an aggressive counteroffensive, heavy cycles of chemotherapy, targeted radiation beams, and immunotherapy. Most cancer cells die at this stage, unable to survive in the unfamiliar chemical environment of a new organ. But you are resilient. You secrete specific growth factors that manipulate the local stromal cells, forcing the new environment to adapt to you. You begin to divide again, forming a secondary tumor site, a metastasis. The host starts to feel the systemic weight of your multi-front war. They experience unexplained weight loss, cachexia, chronic fatigue, and organ-specific failures. If you are in the bone, the bones weaken. If you are in the brain, neurological deficits begin. Oncology teams launch an aggressive counteroffensive, heavy cycles of chemotherapy, targeted radiation beams, and immunotherapy.
Your secondary colonies have taken over. The liver, lungs, and skeletal system are completely saturated with your architecture. You consume the vast majority of the body's energy reserves, leaving nothing for the vital organs to function. The host's blood chemistry collapses. Metabolic acidosis sets in. The kidneys stop filtering waste, and the heart slows to a weak, irregular rhythm.
The monitors in the intensive care unit drop into a long, continuous tone. The host passes away. Because you are entirely dependent on the host's oxygen and warmth to survive, your own empire goes dark within minutes. Your immortal cells finally stop dividing, breaking down alongside the system you conquered. But back in the outside world, inside another normal living human tissue wall, a random copy error occurs during routine cell division. A single strand of DNA snaps. The cycle begins again.